And next talk is devoted to the facile synthesis and structure of germanium nanoparticles and their interaction with body cells. And we kindly invite Professor session1Ersoy from Queen Mary University of London, United Kingdom. And are you ready, Professor Ersoy? Are you ready?
Yes, I am ready.
Okay, and we kindly request you to give your talk within 10 minutes, if possible. We kindly request you. Okay, we have a limited number of people and time, please, because the next session also will start here. Okay, next parallel session. Okay, 10 minutes, and we kindly ask to complete your talk, please, if possible.
Yeah, I have to upload my presentation.
Please, it's okay. Yes.
Yes. Okay, I'm starting.
We can start, please. Yes. Yes, please.
Good afternoon, everyone. My name is session1Ersoy. I am actually a PhD student in condensed matter material physics at Queen Mary University of London. I am in my third year. I'm at the beginning of my third year. Okay, today I'm going to talk about a part of my PhD project. As you can see from the title, it will be about synthesis of germanium quantum dots and its interaction with body cells. First of all, I would like to thank organizing committee, especially Professor Rauf Jafarov, for giving this opportunity to talk here. Now I would like to share my outline with you first. I'm going to introduce my, in the interaction part, I'm going to talk about nanoparticles and what are they and why they are special in terms of quantum confinement effects. Also, in the next part, I'm going to talk about the experimental section, where I will share details of synthesis part. And also, after the synthesis, we did characterization to understand what is our materials. And after that, I will talk about results and discussion and give a sample, example of biological applications and future studies. Okay, next slide. Yes, nanoparticles are generally, spherical quantum dots. Also, there are triangle and square one, but basically spherical quantum dots. They are usually semiconductors, and their electrons is confined in 3 dimensions. Because of these features, they have a very efficient light emission. Also, we can adjust their optical properties by changing their size. They are tunable particles, and because of that, they have very high potential usage for lots of applications. As you can see, it's the first application of gold nanoparticles in mice. They have different kind of external spectra. And also, this is the picture of from our lab. We are synthesizing nanoparticles, and when the size changes, their light change and their appearance change. You can see that this is also a picture of commercially available cadmium selenide quantum dots, and they are very bright. And most of the people are using their light efficiency features. As I said before, they are tunable. When we change the nanoparticle diameter, as you can see this picture, this is the cadmium selenide particles. Their absorption spectra are changing, and we can use specific applications for that. Also, we can change nanoparticle surface. Also, it enables us to play with their absorption spectra. Okay, why we are using germanium quantum dots? Because for elements of the periodic table, they can provide low toxicity. That's the reason many people are using semiconductor materials like silicon or germanium. Silicon is the first element that scientists discovered their optical properties, and silicon is used a lot. And this is the picture of silicon. And also annealing silicon particles, we can change their absorption and different features as well. Why we use germanium is, first, I think germanium is not hasn't been studied a lot. We don't know a lot of information about germanium. And also, they have very narrow band gap. It's easy to excite electrons in first state and to excited states. Also, germanium nanoparticles has very large bore radius when we compare it with silicon. It's analogous as when we're getting reduced the size, we are observing more optical properties of germanium. Also, germanium has non-toxic and very easy to produce. That's the reason why we are using germanium particles actually. There are two methods of main production methods. First of all, top-down method, which is this. In this case, from a bulk material, we can use stain etching or electrochemical or vapor deposition or laser ablation. We can break down the particles, and we will able to obtain small particles. Or the other method is bottom-up technique. In this case, atoms are nucleated in the form of different routes, such as colloidal synthesis, chemical vapor deposition, solder, and so on. In order to produce germanium particles, there are many methods. I choose some of them. For example, solder methods like stain etching, laser ablation, chemical vapor deposition are some techniques that we can produce quantum dots. I tried most of them. But I'm not using them. Why I'm not using? For example, in sol gel method, we have to wait for a long time, and also germanium nanoparticles embedded in silica. Not, for example, after the synthesis, I'm not able to obtain as freestanding germanium particles, and also height required. For example, in stain etching method, it's very acidic and dangerous and chemical. We have to use very dangerous chemicals, and it's also harmful environment. In terms of laser ablation, it's not providing us to control the particle size. In laser ablation, we have our bulk material in a vial. We are sending continuously laser beam on it, and we can break down particles, but it's not efficient to have a stable particle size. Chemical vapor deposition also, there is a picture here from the lab, and we have to go up to high temperature like 500 Celsius degree, and we need vacuum environment, and complicated setup is required. What we are using in our lab, it's very easy and facile method is colloidal synthesis. Colloidal synthesis is size control is important. We can control size precisely, and also various surface termination is possible over the core of germanium nanoparticles. And also it has different sections like shrink method, high temperature method, and benchtop method. These two is also, as you can see this picture, very complicated setup is required. It's difficult. For example, if there is a leak, it's difficult to find, and transfers the chemical is also difficult. High temperature also, it's expensive method. What we are using here is benchtop colloidal method. It's very easy, and you can do it in room temperature synthesis, and very short time method. In 3 hours, you will be able to produce your particles, and also, it's freestanding, like core nanoparticle you can have. Okay, let's have a talk about the detail of our particles. We are using two methods to produce germanium nanoparticles. First method is colloidal synthesis. I call it CS1, colloidal synthesis 1 route. In this case, what you need is 3 necks, bottle like that. First, we are putting here is like ethylene glycol and the other chemicals. I'm not going details a lot. And also, we are using sodium borohydride as a precursor. We are just injecting it, and we are dropping sodium borohydride in different rates. During the process, after 1 hour, it changed the color, and after that, I'm able to obtain particles like powder in this image. There are powders on the glass slide here, and I can have nanoparticles in ethanol or hexane, like in liquid form. The second method is also this is the aqua method. Also, it's benchtop. It's very easy to synthesize, as you can see from the picture. Like we have our mixture of our samples, our elements, like here, And I am using here is magnetic stirrer in 60 centigrade degree, like a little bit up to room temperature, and I am stirring it for about an hour. And after that, I'm able to obtain my particles like this. Here in powder, also I can suspend in methanol in liquid. Okay, let's talk about a little bit results. First of all, in the first method, in order to understand what we produced, I took transmission electron microscope pictures. As you can see from the picture here is spherical particles. Size distribution is about 3.6 nanometer. Also, I took Raman spectra. What is it? To understand what is it actually. This is the very nice asymmetric peak of germanium peak, which is around 290. It tells us, this asymmetric peak tells us it's the nanoparticles. If it is not asymmetric, we can say it's hard to believe that it's nanoparticle actually. Also, we are seeing from the picture basically. And also, I run XRT experiment. This peak is germanium peak also. In the second method, also I took TM images of my particles. In this case, nanoparticle size is around 4. And also I took another Raman spectra to understand what is it actually. It's also another asymmetric peak. This here is the XRT peak of the XRT spectra of our particles. Also, it suggests that germanium is in the core. Here are some optical analysis of our particles. What I did is I also took absorption spectra first. This is the absorption spectra of first method, and this is the photoluminescence spectroscopy of our particle. It's around 700. I'm not sure I'll be able to read or not, but yeah, it's around 700 nanoparticles. This is the image of light is coming out from our particles. This is the second photoluminescence spectra of my particle for the second method. Okay, after that, also I showed that I'm able to change the particle size and also surface of the particle by annealing the particles. It's after 1 hour annealing in hydrogen-argon gas flow. This is the first. Is the red one? The black one is prepared sample. This is the Raman spectra. There is germanium hydrogen here. Germanium hydrogen peak is here. But after the annealing, this hydrogen is removed, and there is a peak, which indicates that it is the germanium carbon peak. It suggests that after the annealing, my particles attach with carbon atoms, surrounded with the carbon atoms, and it's easy to apply any medical, any biological precursor like ligands and other stuff basically. Yeah, after that we apply our particles to our body cells. We use HeLa cells. Here you can see in different channel we can use emission spectra of the particles, and in different channels like 405 nanometer, 488 nanometer, 561 nanometer channels, you can specifically tune and see the particles interaction with the cell. Also, we done in order to understand toxicity effect, we have a look. We did Muse test, which is basically what it means is that we put our particles in a cells. Actually, its intake is doing with the cells, and also we had a look. Okay, how many cells are died after intake? We did a comparison here in 24 hours, and during 3 days we did some measurements. This one is this blue one is germanium particles. This is the red one is commercially available cadmium selenide particles. As you can see, in the first day, it's around the same. This the green one is the 10 times more concentrate germanium particles. In second day, after 48 hours, our particles is when we compare it with commercial one, it's very nice actually. They are not killing cells. In the third day, as you can see, even the 10 times more intense particles are not that much killing the cells actually. They are not that much toxic. What I'm saying is, okay. As a conclusion, we have carried out synthesis, and we understand that we are obtaining, crystalline germanium nanoparticles, and also we did some, luminescent image experiment with, HeLa cells. Also, we test toxicity, germanium nanoparticles, comparing with carbolic coated, cadmium selenide and zinc sulfate commercially available nanoparticles. We found out that our particles are the least toxic when we compare it with these two. After 3 days, it suggests that our particles can be used in clinical imaging and drug screening trials. And I would like to thank my supervisor, Andres Spikin, and my colleagues, and also our sponsors, Turkish Ministry of Education, Primary and Diamond. Thank you for your patience.
Thank you very much, Professor Ersoy, and we are very grateful to you for your interesting talk. And any question from the online connections or from our colleague in Baku State University? Any questions, please? Any questions?
Can you go back 2-3 slides back? Can you go back 3 slides? Go back. Go. Okay, stop here. But no. Next. Okay, but here you discuss about maximum around 1,050 centimeters. Okay, but I think maybe it is some interference picture. No? Are you sure that it is something? But this peak, very like interference peak between. It is interference peak.
This peak, right? You are talking about germanium carbon peak.
Yeah. I am talking about this peak. But
it's not that much. It's not that much sharp peak, but when I search from the literature, there is a carbon germanium peak exists around 1450 nanometer around this region. But yeah, as you can see, it's not that much. It's not precise saying that. But the basic thing is that what we can say that.
So that's why it is not interference. Okay, I understand.
Yeah. But what we are basically saying.
Thank you very much.
Yeah, hydrogen. Okay, thank you.
Thank you. Okay. If there is no another question, then let's applaud Professor Ersoy. Thank you very much.
Thank you.
Thank you very much. Okay.